RNA vaccines, DNA vaccines, virus-like particles, these were all experimental vaccines at best. They had been used in veterinary medicine, but in clinical practice for human population, these vaccines have not been tested yet. Today, the medical fraternity has been catapulted into the future in regards to the experimental vaccines which are going to come up. Today, before addressing the vaccine strategy for COVID-19, I would like to speak about the basics of vaccines and what kind of response they elicit, the different types of it, and finally, a word about the WHO approved candidates for vaccines. Vaccine is defined as a biological preparation prepared from live organisms to enhance immunity against a disease, to either prevent it or to treat the disease. It is either administered in an injectable form, orally, or intranasal route. Be it a vaccine or a disease, when the human body is exposed to a pathogen, the antibody response and T-cell response is something which is represented by the initial immune response. This response lasts for around 7 to 21 days, following which there is a period of protective immunity. When the body is exposed to the disease causing pathogen once again, the immune response is spiked, but the clinical presentation becomes inapparent or very mild. The basis for the immune response is divided into two forms, which is either humoral immunity or cell-mediated immunity. Humoral immunity is mediated by B cells. When an antigen or a pathogen is exposed to the B cells, they produce already an innate amount of antibodies, and the B cells itself transforms itself into a plasmacyte, which helps in mounting a response against the antigen. If the antigen has been neutralized, then this plasma cell eventually becomes a memory B cell. However, in order to mount a more robust form of immune response, the B cells can actually recruit the T cells, and T cells represent the cell-mediated immunity. The T cells release the T killer cells, which are helped with the help of the T helper cells. In our situation, when the body is exposed to a viral pathogen, which is restricted to a cell, be it the B cell or the plasma cell or the macrophage itself, they are marked by an MHC1 binding protein. And this helps in the T killer cells to identify it and destroy that cell. The T helper 1 cells predominantly help during viral and bacterial infections, whereas T helper 2 cells help in parasitic form of infections. In order for a vaccine to mount humoral and cell-mediated immunity, it needs to be designed in a particular way. The live attenuated vaccines successfully induce both the humoral and the cell-mediated immunity. However, the subcomponent vaccines are capable of inducing only the humoral immunity. This is where lies a challenge in deciding the form of vaccine. So in order to decide what form of vaccine has to be used for a patient, we need to go back to the history to understand a bit about what the different types of vaccines were and how they were actually discovered. The concept of immunity was there long before it was discovered by Edward Jenner. By the Buddhist monks, the form of immunity was discovered by using snake venom. They were successfully able to ward off immunity by drinking a particular form of snake venom. However, the concept of inoculating a particular form of pathogen was practiced in Central Asia, China, and Turkey. This practice was known as variolation, where dried pustules of smallpox were inoculated from healthy individuals, from diseased individuals, or recovering individuals to healthy population. This was practiced between 15th and 17th century. However, the word vaccine is attributed to Edward Jenner, who inoculated a cowpox virus to a child in order to prevent smallpox. How he managed to do that is speculated that when he acquired the cowpox virus from a pustule of a cow, of a woman who was attending cows, she might have been infected with smallpox as well. So that way, he was able to ward off smallpox in a child. Later in the 19th century, Louis Pasteur was able to demonstrate how a virus, how a harmful disease-causing pathogen can be attenuated in lab conditions. He demonstrated this in chicken and in animals, and he was able to successfully develop a vaccine for rabies in the following year. When Salmon and Smith were able to demonstrate that vaccine can be developed not just from live organisms, but also from killed organisms. They named it an inactivated vaccine. In the 20th century, a line of new concepts in vaccines were developed. One of them was called as toxoid, and the following one was named as conjugation of polysaccharides to mount a particular form of response. Eventually, in the 21st century, what we are witnessing today is genetic engineering and genomic analysis. So to summarize the history, we have live attenuated vaccines, which are in the form of BCG and oral polio vaccine and MMR. Inactivated vaccines as injectable polio vaccine, hepatitis A, and the pneumococcal vaccine. The subunit vaccine, which is in the form of hepatitis B. Toxoid vaccine, which is diphtheria, tetanus, and pertussis vaccine. The conjugate vaccine is an example for that is Hib, meningococcal, and pneumococcal vaccine. Another component of a vaccine which is sold in a vial is the adjuvant and the preservative. An adjuvant is something which can enhance the immune response of a native vaccine preparation. Aluminium is a very commonly used adjuvant, and aluminium salt along with monophospholipid A is used for human papilloma virus vaccine. The MF59 is used for the H1N1 vaccine. Preservatives are generally what ensure sterility of a vaccine. An example for that is the thiamersol, which is used for multi-dose influenza vaccine. How exactly these vaccines are prepared? A live attenuated vaccine is grown with the help of a disease-causing agent. Growing this agent in a suboptimal condition would make sure that the disease is attenuated in the following generations. The progeny which is finally grown is nowhere close to the virulence or the pathogenicity of the disease-causing pathogen. Finally, this preparation is purified, formulated, and sold as a vaccine. However, this form of vaccine has its own problems. For example, the oral polio vaccine is capable of reverting back to its original pathogenicity and causing paralysis in children. Hence, there is the surveillance program by the WHO. The inactivated vaccines are produced when a disease-causing agent is inactivated with the help of thermal or chemical preparation. This eventually is purified and sold as a vaccine. The later development was the recombinant subunit vaccine, which is developed especially in the hepatitis B vaccine, when the surface protein, the HBS component, is specifically extracted and inoculated into an E. coli vaccine. And this particular subunit is expressed so that the E. coli is able to grow this particular subunit. That is now purified and sold as a vaccine. This type of vaccine is able to mount a humoral form of response. Finally, the toxoids. Unlike the previous diseases which were caused by the pathogen itself, there are certain kinds of diseases like diphtheria and pertussis where the actual disease-causing organism is not as dangerous as its toxin. Hence, the toxin is isolated, chemically treated, purified, and finally sold as a vaccine. Conjugate vaccines are a newer form of vaccines where polysaccharide component cannot induce a good amount of humoral or cell-mediated immunity. Hence, if this is conjugated with a particular form of protein, that is successfully able to mount both humoral and cell-mediated response. An example for this is the Haemophilus influenzae, pneumococcal, and the meningococcal vaccine. Now in the 21st century, thanks to the genetic engineering and genomic analysis, we have RNA vaccines, DNA vaccines, and virus-like particle vaccines. Now that is where I'll be speaking in the second half of my presentation. However, another important part in understanding whether a vaccine is useful or not, there are two concepts here, the vaccine efficacy and the vaccine efficiency. Vaccine efficacy is the attack rate, which is calculated with the help of how many unvaccinated people in a population can get the disease in comparison to those who are vaccinated. And it's a mathematical calculation. At best, the vaccine efficacy is an in vitro study, and it is equated to the concept of herd immunity. However, what is more important in understanding is the vaccine effectiveness. A vaccine effectiveness looks at what kind of outcome did the vaccine actually cause. How many patients after receiving a vaccine were actually hospitalized and how many of them actually died. So this is more of an in vivo analysis. Finally, when we speak about the herd immunity basically represents that parameter when a disease transmission is interrupted to an unvaccinated or the susceptible population by a vaccinated crowd. It is not surprising to understand that in coronaviruses, the herd immunity lasts for maybe 1 or 2 years, not more than that. The reasons for why this happens is still not known. So coming down to the basis of vaccine development. In order to develop an effective vaccine, it is important to understand what the structure of the coronavirus is. The SARS coronavirus 2 has a nucleocapsid structure, a genomic material which is RNA and which is positive stranded. It is covered by the N protein, which is represented by the nucleocapsid, and which is further covered by the envelope. The envelope consists of the spike protein, the envelope protein, and the membrane protein. One of the more effective ways of developing a proper vaccine would be the whole cell antigen, which means that all the elements of the virus have to be considered to make a vaccine. So the best form of vaccine would be a whole cell killed vaccine or an attenuated vaccine. However, this form of vaccine requires longer duration because it requires more stringent screening, and finally the progeny which is developed should not have the same kind of pathogenicity. Hence, the world is not exactly ready to get a whole cell antigen vaccine yet. Hence, the direction of developing the vaccine for most of the vaccine developers is towards component vaccines. They have targeted specific parts of the envelope, which includes the spike protein, the envelope protein, and the membrane protein. The spike protein is a particular direction where most of the research is heading towards. A word about what exactly the genome of the SARS coronavirus 2 holds and what exactly does it translate into. What is represented on the top is the actual length of the RNA. The RNA is 30,000 base pairs in length. It consists of open reading frames, which are numbered from open reading frames 1 all the way down to open reading frames 10. However, what is more important at this point of time is the analysis of the S, which is the spike protein. What is shown in the red and what is shown below is the two subcomponents of the spike protein. The spike protein consists of two subunits, which is the S1 and the S2 subunit. The S1 subunit has a specific component called the receptor binding domain, whereas the S2 segment consists of the transmembrane domain, the heptad repeats, the cytoplasmic domain, and the fusion peptide. Now, I have taken the liberty to actually give a diagrammatic representation for the subunits. The S1 subunit looks like a cap, whereas the S2 subunit looks more like a conical structure. The S1 subunit consists of a receptor binding protein, which is the one that has affinity towards the ACE2 receptor. Now, in comparison to the previous coronaviruses, this is the component which has the most variance in comparison to the previous ones. It is different and it does not have any form of resemblance with the previous SARS-1, MERS, or the animal coronaviruses. What happens is once the subunit 1 has been has its affinity towards the ACE2 receptor, the S2 is somehow revealed and it is exposed to the host cell. This consists of a cleavage site with the help of which the S1 subunit is able to separate off, and the fusion protein somehow forms a bind with the ACE2 receptor. Based on some of the genomic analysis, this is a diagrammatic representation where on the top we see the host cell and on the bottom we see the virus cell. What is represented in blue color on the left hand side is the pre-fusion spike. The S1 component is shown in blue, whereas the S2 component in green. The affinity of the S1 is towards the ACE2 receptor, and the TMPRSS2 gene is implicated in the binding of the S1 to the ACE2 receptor. Once the binding is complete, there is cleavage of the S1 subunit, and finally the S2 subunit binds towards the host cell and it forms a hairpin intermediate, and finally the binding between the virus and the host cell becomes significantly stronger. This way, the virus is able to transfer its genetic material to the host. So now that the understanding of the S protein is, the understanding of the S protein actually leads to the development of multiple vaccines. As of now, there are more than 80 projects which are ongoing, and most of them are either targeting the whole S protein or components of the S protein. The receptor binding domain is where most of the subunits are being, subunit vaccines are being developed. The N-terminal is another site where the vaccine is being developed towards. And finally, the S1 subunit has shown some promise when it came down to the MERS coronavirus vaccine. The FP, the fusion peptide domain, is another site where vaccines are being developed for. Finally, when the envelope or the membrane proteins were considered, they do not hold much immunogenicity to make a promising vaccine. However, there are certain problems when considering the spike protein. The actual spike protein is covered by the N-linked glycans, which can successfully evade the immune system. The receptor binding domain also holds a very closed form of conformation, which is hidden from the immune system. Even though a vaccine can be developed, the problem is that it can be narrow spectrum, limiting it only to the COVID-19 disease. Hence, a more robust form of a vaccine needs to be developed. S2 component also needs to be considered in this form of virus. So what are the WHO approved vaccine candidates here? We have 5 vaccines which have been approved and which are in various stages of development, and 71 others in preclinical evaluation. The inactivated vaccines are being developed by the Beijing Institute, which use the whole component, and not only the Beijing Institute of but the Serum Institute of India, along with the Codagenix company, are on the way to create a live attenuated vaccine. A new form of vaccine, which is the non-replicating viral vector vaccine. It uses the adenovirus type 5, which consists of only the spike protein, which will eventually present the spike protein to the human body once inoculated. This India, along with Dr. Adrian Hill from Oxford University, have used chimpanzee virus to present the S protein. Another experimental virus, which is the DNA plasmid extrapolation virus, is also vaccine is being used. A particular form of plasmid, which consists of the spike protein and other forms of antigenic material, are inoculated into the E. coli, which will be grown in a culture medium. And finally, when the plasmids are developed, they are purified and they are presented to the human body, so that they will be taken in by the humoral and cell mediated immunity to present the spike protein. The lipid nanoparticle vaccines are being developed by Moderna. This is in phase 1 of clinical trial. We do not know exactly what the encapsulated mRNA vaccine would be, but these are the possible two types. The non-replicating mRNA, which would probably be taken up by the cell and be presented as an antigen, or a replicating form of mRNA, which would reproduce within the cell and would be able to present to the cell-mediated immunity as well. To be honest, the COVID-19 vaccine is a form of race. It is a race between various countries, India included, pharmaceutical companies, and private organizations. There is a multitude of information right now where NCBI virus and GSI-8 have released the virus genomic sequence so that vaccine developers have information to use. The clinical trial designs have also been changed, and now there are multiple companies which are actually making the vaccine. However, with adaptive trial designs, we have certain set of problems. This is the chronology of how a vaccine should ideally be going through: the experimental vaccine, the testing of immunogenicity, the manufacture, toxicological analysis, and finally the phases of the phase 1, phase 2, and phase 3. What eventually is happening today is there is skipping of one or particular steps or combination of phase 1 and phase 2, phase 1, phase 2, and phase 3 altogether. Generally, this whole staging takes nearly 15 years for a particular form of vaccine to develop. When this process was actually hurried up in the past, we had problems. And respiratory syncytial virus is an example. In the 1960s, when it was introduced to children, there were enhanced form of diseases. A similar form of problem came up during the swine flu vaccine and antibody enhancement in the recently dengue virus vaccine. This also poses for certain organizational problems, which is the monopoly by the pharmaceutical companies, the eventual cost of the vaccine, maintenance of cold chain, and the actual vaccine efficacy. And finally, a word about what the vaccine, BCG vaccine, is actually implicated in the impact of COVID-19. Recently, there were multiple studies which came up which showed that BCG vaccine holds protective effect on COVID-19. As of now, we do not know what the role of BCG is, and there are two major trials which are going on, which are BCG Corona and the BRACE trial. We still need to wait for the results of these trials. As of now, there is a lot which we still do not know, but we need to wait for answers which would be appearing in the coming 1 year or so. These were my references, and thank you very much.